So here we'll talk about primary amoeba meningoencephalitis. It is caused by Naegleria fowleri, which is a free-living amoeba, so called brain-eating amoeba. So epidemiology, it is rarely seen, but it can be most commonly seen in USA, greater than 100 cases per year, and in Rajasthan and Kolkata, greater than 20 cases. So what is happening here? This Naegleria fowleri exists under three forms. One is amoeboid trophozoite, which is very important, and second is flagellated trophozoite, third is cystic form. So this amoeboid trophozoite is the diagnostic and the infective form you have to remember in this disease. So this is responsible for the infection. So here you can see the structure of the amoeboid trophozoite. You can see the food vacuole, cytoplasm, karyosome, nucleolus, and the important thing you must remember in amoeboid trophozoite is lobopodia. This word is important, lobopodia, right? Under flagellated trophozoite, you can see the flagella, and the cystic form, you can see this thick double-walled cyst wall. So these are the three forms of Naegleria fowleri, and the amoeboid trophozoite is responsible for the infection, and also you can see it under the microscope. When you collect the CSF fluid from infected person, you can see it under the microscope. Into pathogenesis, so what is happening here? The free-living amoeba or the amoeboid trophozoites of this Naegleria fowleri are present in one swimming pool, and a person swims there. While swimming, he accidentally inhales this amoeboid trophozoites of this Naegleria fowleri. They penetrate the nasal mucosa, they infect the olfactory nerve, and then through the olfactory bulb, through the olfactory nerve, it reaches the brain and causes primary amoebic meningoencephalitis, that means infection of the brain, and the person dies less than 2 weeks. You see the clinical features. After 1 or 2 days after the swimming, the person can seem to develop this fever, nausea, vomiting, anorexia, that means inability to eat, and change in smell and taste, and also neck stiffness is present. Positive Kernick's sign. So the patient will have fever with chills and also neck stiff. As the disease progresses, you can also see the CNS manifestations like confusion, ataxia, hallucinations, lack of attention, seizures, and in 98% of the cases, death is confirmed within 2 weeks. Into lab diagnosis, if you do the CSF cerebrospinal fluid analysis, you can see these pulses which are greater than 20,000 mul in number, and you can see this thick purulent CSF. Why? Because it is filled with the pulses, right? And also microscopy, again as I said, you can see this amoeboid trophozoites which are better seen in case of phase contrast field. Phase contrast microscopy, you can better see this amoeboid trophozoites. Looks something like this with the lobopodia. Histopathology using H&E stains, the cytoplasm is stained blue and the nucleus is stained pink. And if you do the culture, see, for the culture, I want you to imagine a plate where you have this E. coli. If you drop one of the Naegleria fowleri into it, it just eats all the E. coli and it makes a sign which is called trial sign. That means you can clearly see this Naegleria is feeding on the E. coli. It's important to remember this trial sign, and also the thick purulent CSF which is filled with pus cells, and neurological symptoms and neck stiffness. So olfactory nerve infection, amoeboid trophozoite, and the lobopodia. These are the things you have to remember. The treatment you can give to this disease is amphotericin B, but the death is almost confirmed. For less than 2 weeks, you can see the person dying. So bye.